going to introduce our first speaker of the afternoon in the track A. And as I mentioned before the break, this is currently my favorite project that is happening on top of Spark. And Jeremy, who's the speaker, is a fellow at a really unique research lab, which I hope he tells you all about.
All right. Thanks a lot, Andy, for the introduction. I'm very excited to be here and tell you about how we are using Spark to map and manipulate the brain. So I am a neuroscientist, and I want to understand how the brain works. And what I'm showing here is a very kind of high-level picture of what the brain does. On the left are stimuli. These are things out in the world that we need to encode: visual information, auditory information. And on the right is behavior. These are the actions that we take, our motor plans. And the brain is responsible for basically everything that happens in between. The brain consists of a bunch of individual elements. There's little circles here meant to represent neurons. Neurons are organized and connected to one another in complicated ways. They communicate with one another. Different neurons process different kinds of information, and they work together to enable our brains to represent information in the outside world and act on it in a meaningful way. I think you'll agree with me that in order to understand a system like this, we really need to look at all of it at once. We need to be in control of the stimuli. We also need to be in control or monitoring the behavior. And ideally, we'd be able to look at what every single neuron is doing. Now, this is a challenge because there are a lot of neurons. I'm showing here a sort of logarithmic chart of how many neurons are found in a bunch of different animals, all the way from the very unintelligent snail, which has about 1,000 neurons, all the way to humans, which have about 100 billion. So I'll tell you right now, we are nowhere near close to being able to record the activity of every single neuron in the human brain simultaneously. That's still a ways away. But due to some very recent exciting advances in technology, it's now possible to record from many, many neurons, in some cases all of the neurons, in some of these simpler animals. So what is that technology? In the early days, a lot of efforts to record from neurons depended on dropping little wire electrodes into the brain in order to listen to the electrical activity of neurons. And by clumping some of these wires together in what's called a multi-electrode array, we can record from, in some cases, 10 to 100 neurons simultaneously. So that's pretty good, but it's still nowhere close to all of them. A big advance came from the idea of using not electrical activity to record neurons, but instead using light. So the idea here is that we genetically encode or engineer animals so that their neurons light up when they're active, and then using a combination of lasers and optics, we can produce images that show the activity of neurons, visualize the activity of neurons over very large fields of view. And one particular method called two-photon imaging, we can get up to 100, in some cases 1,000 neurons simultaneously. So that's a lot, it's still not quite there. Another very recent and very big advance came from a method called light sheet imaging, which was developed and used by my collaborators, Philip Keller and Misha Arens. And the idea of light sheet imaging, at least quite simply, is to excite with a laser not a single point in space, but instead excite along an entire beam. And intuitively, this allows us to cover a much larger volume of neural tissue and record from many more neurons simultaneously. Of course, the details are a lot more complicated. So with light sheet imaging, we can now measure the activity of about 100,000 neurons simultaneously. Now, one big caveat to these methods that use light is that we have to somehow find a way to look inside the brain. So most brains, like ours, are inside a skull, so we can't just look inside them. But we have access to a very cool model system, which is the zebrafish. So a zebrafish, which looks like this, it kind of looks like an alien, in its larva form, it turns out the zebrafish is transparent, which means that if we put it under one of these microscope laser setups, we can essentially look through its brain and image activity of its entire brain simultaneously. So here is a movie showing what the results of such imaging look like. This is from a paper that was published just earlier this year by Misha and Philip. So what we're watching here is neural activity at a very high resolution across the entire brain of the zebrafish. multiple frames collected, or one frame collected every second. All of these different little planes are different cross-sections through the zebrafish's brain. There are about 50 of them, so this is a three-dimensional stack. And we're seeing how neural activity evolves over both space and time in the brain of this intact animal. And this was, when it was published, the first example of really monitoring every single neuron in a brain. Now, you can imagine that these data get pretty big. And in fact, these experiments are yielding data sets that are much larger than any collected in neuroscience before. So the kind of images I just showed you yield data sets that are both spatial and temporal at every moment in time. We have these multiple cross-sections. Each cross-section is a very high-resolution image. And in the course of about 30 minutes of an experiment, these images end up generating about a terabyte of data. Over the next couple years, this number is only going to grow because we're trying to do these experiments faster imaging rates, and also look over much larger volumes, eventually maybe to do this at the scale of, say, many brain areas of a mouse. So the data sets are really big. They're also very complicated. These data are spatiotemporal in the sense that at every location within one of these high-resolution images, we have a time series describing how neural activity at that location is evolving over time, like those 4 example traces shown here. Now, there are a number of analyses that we might like to do to try to find structure, or sort of understand the structure in these neural data. This includes simple things, like shown below, summary statistics, where we just want to compute the average activity of every neuron or the standard deviation. We want to do regression-like things, where we try to predict neural responses as a function of things in the outside world, or the animal's behavior. We also want to do analyses that look at structure across the entire data set simultaneously, things like dimensionality reduction and clustering. Another key point about these analyses is that when just given a data set, we never quite know at the beginning what analysis to do. It's a very exploratory process as we've come to understand these data. So in thinking about how to analyze data, neural data at this scale, there were a few things we decided we needed. We need speed. We need to be able to do quick analyses on these increasingly large data sets. We needed to do iterative calculations, because a lot of the models that we want to fit involve repeated iterative estimation. And we also wanted to be able to do things interactively. Again, we don't always know the right analysis, so it's very useful if we can try a bunch of different analyses on a single data set and try to understand, sort of in an exploratory way, the structure in the data. So about 99% of neuroscience works with a very simple workflow, which is to collect the data, say on the microscope, and then analyze it in MATLAB on a single machine. But we were pretty confident this was not going to be a scalable solution. So we were very excited to realize that our needs and our interests in coming to analyze these data lined up very nicely with what was offered by Spark. So we built a new workflow for analyzing these data. We implemented Spark on the cluster we have at Genelia. And in this new workflow, our data come directly from the microscope to the cluster. We then use Spark to do a variety of different kinds of analyses on them and spit out results in a variety of different formats, images, JSON, other things, that we can then visualize or examine in various downstream applications. The analyses themselves we've been building as part of a library that we've been writing. This library has a combination of analyses and algorithms for a variety of sort of general machine learning tasks, and then also problems specific to neural data analysis. In that sense, it's quite complementary, I think, to ML-base. A couple things to say about it. One is that we have a mix of both low-level algorithms and also more kind of high-level analyses. And we've written sort of very simple scripts that let us chain together different kinds of analyses. In the example I'm showing here, we can quickly parse a neural data set and then do some kind of regression analysis, and then in the next step, use the output of that to do dimensionality reduction. A lot of this library is written natively in Python because we rely a lot on the numerical routines and optimization routines of NumPy and SciPy. But in a few special cases, we also have Scala implementations. So that's all the technology behind what we're doing. What I want to do now is show you a couple examples of the kinds of experiments we're doing and how we're using these large-scale analytics to find interesting patterns in the brain. So the first one is the example of direction selectivity. It turns out that in basically every animal, all the way from a fly to a human, there are parts of the brain that are specialized for representing motion, visual motion, in different directions. And this might be useful because all animals at some point have to, for example, track prey that are moving in front of them or track a predator that's trying to chase them. So you want to know which way things are moving. Early example of this came from the primate. I'm showing here on the right traces of the electrical activity of a single neuron, and on the left are bars that were moving in different directions. And I think you'll appreciate this neuron responds more to bars moving in some directions than others. But of course, this is just one neuron, and in the zebrafish, we have the ability to look at selectivity to the direction of motion literally across the entire brain. So to do that, we do an experiment in which we present underneath the fish, so the fish can see, bars that are moving in many different directions, and present them in sequence. And here's a movie showing what activity, neural activity, across the entire brain looks like in this experiment. In the upper left, I'm showing the stimulus, which again was just moving bars that came on and off. And in the image, we're seeing in bright white activity of neurons across the entire brain. And what we can notice, but might be a little hard to make out, is that different directions are eliciting different patterns of neural activity. Now, the problem is just looking at this movie, it's not immediately obvious what all those different neurons are doing. There's a lot of rich structure here. So that's where the analysis comes in. I'm going to re-represent these data not as a movie, but as a time series. So for every location within that image, we can look at how neural activity changes over time in response to the 12 different directions shown at the bottom. In these traces, the stimulus was off during the black period and on during the gray period. So most of these neurons respond when the stimulus is presented, and they do so more for some directions than others. So in order to make sense of all of these time series, we do a fairly simple analysis in which we take each time series independently and we fit a parametric model to it. This model has two components. One is meant to capture the temporal shape of the response, and we assume that that's common to all directions. And then we also assume there's a tuning function which describes how that neuron responds to some directions relative to others. This gives us a preferred direction for every neuron, as well as the width of its tuning. How much does it care about that one particular direction? And in essence, what we've done is assign a color to every single time series. That color is the preferred direction of visual motion. And I'm showing here for all of these time series what those fits of the model look like. So because we can do this in Spark very quickly on about 68 million time series, we now have an ability very quickly to take the kind of movie that I just showed you and generate a map of direction selectivity across the entire brain. So literally, in just a few seconds, we can generate images like this. So this is a map of direction selectivity, really for the first time, across the entire brain of an animal. There's a lot to appreciate in this graphic. A few things to draw your attention to is that you can see there are regions of this brain where selectivity to motion, the different colors, are kind of heterogeneous and interspersed, like the area at the top and the bottom, and some areas on the far right, which is the forebrain. There are other regions, especially towards the left, which is the hindbrain and the midbrain, where things are very coarse. There's entire patches of the brain that seem to prefer just one direction. So we think this mixing across the brain of both local diversity in preference as well as this kind of coarse organization might be very crucial to the kinds of computations that this brain is performing in order to represent information about visual motion. Another thing to appreciate is just how dense and sort of detailed these images are. So I'm going to show you what happens if we just zoom in on 5 different patches to show you within each of these regions there's incredible richness and sort of precision of these measurements, that we really are picking out the selectivity of basically every single neuron across this brain. So that example was focused very much on selectivity to a stimulus, to something out there in the world, visual motion. But we're also interested in behavior. So it turns out fish, as well as humans and a bunch of other animals, have a very simple behavioral response when things move. Basically, when something moves, we try to move in order to track it. So for the fish, he tries to swim towards something that's moving. In the case of humans, we track our eyes as we watch something moving around in front of us. So we can study this in the fish by presenting, again, something moving underneath him, and then we attach little pipettes to his tail to listen to muscle activity. So we're actually monitoring the effort of this fish to swim, and we can then relate that to the neural responses. So here is another movie. I'm showing now the activity across the brain during a stimulus which is moving, as shown in the upper left. And in the lower left, I'm showing a trace of activity in the muscles in this fish's tail. So I think what you'll notice is that when the stimulus moves in the upper left, the fish starts swimming, so the black trace goes up, and there's a cascade of fairly complicated neural activity evolving across his entire brain. So again, it's a little hard to know what's going on just by looking at the movie, so we need analyses that can help us understand it. The starting point is to, again, look at a time series. So here I'm showing for the stimulus, which was coming off and then on, black and gray. In white is the trace of the swimming. So this is how much the fish was trying to swim. He swims every time the stimulus comes on. And in blue is the response of a single neuron, which is also responding when the stimulus comes on. But of course, there's not just one neuron, there's hundreds of thousands. So we need a way of describing the dynamics of activity across all of these neurons. So the way we do that is through dimensionality reduction. I'm showing here just one neuron. We start by taking the activity of each neuron and averaging it across multiple repeated presentations. And that gives us a large collection of little short time series, as shown in the lower left. Each of these little time series describes the average dynamical response of a single neuron to the stimulus coming on. We then use principal components analysis to reduce these high-dimensional time series data into a simpler two-dimensional space. So every little time series becomes a point in this two-dimensional space, as shown in the middle. And then really the challenge is not just how to do dimensionality reduction, which we do in Spark, but also how to visualize it. So in this case, a key insight is that each point in this two-dimensional space corresponds to a little time series, as shown here. And in this two-dimensional space, as we move away from the origin, we're changing the strength of the response, but not changing the shape. If we move around the circle of this two-dimensional space, we're changing the shape. So it's very natural to color these points where darkness indicates the distance from the origin and hue indicates the shape, or the shape of the time course that we recover using dimensionality reduction. So by coloring every single little time series this way, essentially we can go back to the brain and generate a map of the dynamics. And that's what I am showing here. So this is now an image of the same brain that was shown in that movie, but we've created essentially a color map of the dynamics of its neural response to this onset of stimulus and swimming. And the key in the lower left shows how different colors map onto different shapes of response. The key thing to appreciate is that there are parts of the brain, like shown in green, where the response is earliest. These are responding basically as soon as the visual stimulus comes on. Then there are other areas, like the one shown in blue, that are responding a little bit later, and we believe these are involved in the generation of the motor output. There are also responses, like those shown in red, which respond not while the stimulus is present, but after the fish swims. And we believe that this idea of responses that happen not while a behavior is taking place, but after the behavior is taking place, may be fundamental in how this fish comes to coordinate its swimming activity. So everything I've shown you here is correlational, in the sense that we measure brain activity while animals do something, and then try to assess the kinds of patterns that we see in the brain. But we'd really like to get at something much more causal. We want to know, how are these individual neurons actually contributing to the computation that the animal is performing? And for that, we needed to take another step forward. And this is very much work in progress, but I just want to give you a teaser for the kinds of things we're doing. So the idea here is to use streaming analysis with Spark Streaming to monitor the activity of neurons in real time, and then use a variety of different methods, using lasers, to go in and manipulate the activity based on what our analyses show us. So I showed you before, this is our basic pipeline. We have microscope, data gets sent to the cluster, and analyzed with Spark. We adapted this to use Spark Streaming to do analysis as it's generated by the microscope and sent to the cluster in real time. And then Spark Streaming returns results that we can serve on a local web server. And through a web app that we built using Meteor and D3.js, we can visualize what the brain is doing interactively and choose to do certain manipulations. In particular, we can select cells of interest and then stimulate them using a laser. So I want to show you how all these different technologies come together in an example of literally what we were doing during one day in the lab. In the image, we're looking at a map, in this case a simpler analysis of neural activity. Neurons that we're interested in are shown in white. And that's me with a mouse clicking on individual neurons and highlighting them. And after I've selected all the neurons that I'm interested in, I'll click a button in the upper right that says fire the laser. And this is going to send a set of coordinates to a laser, which will then go in this case, kill all of these neurons. So we find the neurons we're interested in, and then we kill them. And I know it sounds a bit brutal. And the idea is to see what effect that has on the animal's behavior. Show you that a little bit sped up. This is from an actual experiment. And you can see as time goes on, this streaming analysis is giving us a sort of increasingly clean picture of what the brain is doing. And only once it reaches a certain level of clarity do we choose to do our manipulation. So I'm not going to tell you too many specifics about what this actually does to the brain. The short answer is it does a lot. We find that these manipulations have effects on behavior, and they also have effects on the activity of other neurons. So we've kind of created this playground for ourselves to try to understand it. We're also actively developing new kinds of analyses and visualizations. So I'm showing here an example of a implementation of the k-means algorithm, but an online version of it. So this is a little more fine-grained way of classifying or clustering cells into different categories. So I'm showing here, on the right are the time series of individual clusters, and on the left is a map of brain activity and how it gets organized into these different clusters. The analysis is taking place, again, in real time, so we can watch it as the experiment is going on, and then, again, go in and do our manipulations. All right. I want to end by thanking everyone involved. Genelia, where I work, is a very collaborative place. I want to thank Misha in green and all the postdocs and grad students in his lab. We work very closely on the development of these analyses and, of course, the experiments. My scientific computing team, which was super helpful in getting Spark off the ground and running at Genelia. Also, of course, want to thank the Spark development team. It's been a real great opportunity to use their tools and discuss things with them. My friend Deep from MetaMarkets, who helped me with some of the early implementation. My mentors, Michael and Vivek, and my lab coordinator, Emily. And I also want to say that at Genelia, we are every day generating cool, very big and very complicated data sets, and there's a lot of opportunities for data scientists, either through collaboration or various positions we have available, to work on these problems. Thank you very much.